In the human diet, there are a variety of fresh produce such as fruit and vegetables which are consumed raw and have populations of bacteria on the surface, some of which can be harmful to the human body. The aim of the study done by Leff and Fierer was to distinguish the bacterial communities on the surfaces of a variety of fruit and vegetables at the point of sale. They focused on addressing two questions. The first was how the bacterial community organisation varied amongst the different produce types, and secondly, did the farming techniques, i.e. organic versus conventional farming, have the potential to impact the bacterial communities on the produce surface which is consumed by the end consumer. In this experiment, Leff and Fierer chose a large range of fruit and vegetable samples from 3 of the local supermarkets in Boulder, Colorado, USA. These samples consisted of 11 produce varieties, with 9 having both the organic and conventional labelled produce range purchased as well. The bacterial samples were collected from the surface of each of the produce samples via sterile cotton swabs or sterile water rinse. A number of different techniques, a number of which involved the analysis of 16S RNA, were used by the research teams to determine the differences in the microbial community composition across the sample items and the differences throughout the bacterial community composition between farming practices and produce types.
As
They
Figure
found that
1
there were
shows,
differences of
with the C
taxonomic richness
representing
on the
conventional
organic and
labelled
conventional
produce and
labelled
the U
produce, and this
representing
depended
the
on the
organic
produce
labelled
type itself.
produce. The organic labelled peppers, strawberries, peaches, grapes, and mushrooms had a lower bacterial taxon richness than the conventional labelled counterparts, while the spinach, lettuce, and tomatoes had significantly higher bacterial taxon richness in the organic labelled samples, while Figure 2 displays the different families of bacteria found on the surface of the samples. As you can see, the family Enterobacteriaceae was abundant in all of the samples, with bean sprouts, spinach, and strawberries containing more than 50% of the bacteria found on the surface of the samples. The next abundant families were Bacterial and Oxalobacteriaceae.
Leff and Fry suggest that due to the abundance of the bacterial family Enterobacteriaceae, that this family may play an important role in the ecology of its host and may possibly be related to promoting growth and protecting its host from diseases. Leff and Fry also say that it's difficult to establish the certain factors accountable for the differences between the bacterial communities on different produce varieties. However, conclude that it's probably due to several factors, such as variation in handling, transport, and storage of the produce. Therefore, it was concluded by Leff and Fry that the farming techniques and produce types did have an effect on the diversity and composition of the surface bacterial communities. However, Leff and Fry did state that their findings did raise further questions and that further research is required to answer these questions.